So I would like to introduce you a view of the plant as a microbial ecosystem and try to make a general picture on how we can imagine that microbial backbone of the plant is acting in many properties of the plant. And from there, I'd like to, on a specific topic, which is the one on which I'm doing my research, I'd like to go to some lines on how to improve the microbial backbone. My first thing is that this is a microbial ecosystem, and as long as you don't reach that, you don't know what's inside. Because, for example, I'll be very quick on that. Those plants you saw, they are basically microbial, even within their cells. I don't want to go back too much into the old data, but it's quite clear that the plastids, which are doing the photosynthesis, they are of microbial origin. We know that since the very first trees done in the 70s, that we are placing the blue-green algae and the chloroplast together. So there is no doubt that a plant is basically something with cyanobacteria inside for its metabolism. And of course, there is also mitochondria inside. I don't want to be too long on that, but it means that the recruitment of microbes started very early and leaving scares, such as mitochondria and plastids, very deep into what we call a plant. But of course, my talk will not be on that. I like now to go to the root, not of the problem, but to the roots of the plant, okay? And I will introduce an association which is hardly visible because not only it's underground, but also it's hardly visible by our eye. The mycorrhizal association on which my teams in Paris and Gdańsk are working. This is the idea that within healthy roots, you have fungi. It's very often inside. Sometimes it's covering the root, in which case you can see it, but most of the time you don't see the fungus from outside. And this dual organ is called a mycorrhiza. And the mycorrhizal symbiosis is reshaping the plant. First, in terms of nutrition. The fungus is exploring the soil. You hardly see the filaments here, but there is a lot of filaments emanating from the roots. And this is how the plant is able to circumvent the low concentration of the soil in nitrogen, phosphorus, but also all other kind of resources, water included. Those fungi here are the ones that are mycorrhizal to most plants. They sporulate in soil, so their spores are very inconspicuous as well, and it makes this association not well known. The glomeromycota are mycorrhizal to more than 3/4 of the non-plants. Here you have an example of what sterilization is doing. At first glance, sterilization is, you would hope, just killing the pathogens, but it's also killing the ability of the plant to grow normally. Those onions here, they are 3 months old. Here the soil is sterile. Here it's sterile, and there was reintroduction of the spores of the mycorrhizal fungi. And the take-home message is that without those fungi, there is poor growth. Even for some plants, such as pinus trees, for example, there is no growth when the fungus is not there, which is something hardly possible in a normal life. But because at least one of the species is there. But it's really something that is deeply rooted into the life of the plant, the fact that they manage with the fungi that are exploring the soil. It's very often viewed as a trophic relationship, and it is, because the plant doesn't feed without that. And those fungi, they also strictly rely on the plant. This 80% figure is including other type of mycorrhizae than the one I showed to you, because there is a diversity of fungi involved in that, but all in all, 4 out of 5 plant species, they need a fungus to grow. But there is also protection of the fungus by the plant and of the plant by the fungus, but we don't care the fungus, so we will only discuss the first issue. For example, an overlooked fact is that being calcicolous, being able to grow on calcium, is a feature that is largely driven in many plants by way of the fungus. Let's take eucalypts, which are growing on both limestone and non-limestone soil. You can see that when they are mycorrhizal, the soil without limestone or with limestone, they grow nearly as well. They simply grow a bit less well when there is calcium, but it's really not a plant that is bothered by calcium. Now, when it's not mycorrhizal, you divide the growth by 2 when the soil is without limestone. When the soil is with limestone, you divide the growth by 10. And that's it. Most of the calcium is is sent back to the soil by the fungus, which is crystallizing oxalic calcium around its own filaments, making the calcium not bioavailable and making a protection. That's the raw story. It's a bit more complex. But for most plants that grow on limestone, they just are protected from the toxicity of calcium by way of the fungus and the fungal partner. Of course, there is protection against diseases, and you could imagine that it's a local protection where the plant, the aggressors of the root, they meet the fungus and the roots are protected by the fungus. But the truth is that the protection is systemic. Let's take some tobacco plant with or without mycorrhizae, with mycorrhizae or without, correctly fed with supplement of nitrogen and phosphorus, so that we have no artifact on nutrition level or physiological status. And let's challenge them by gray mold agent, botrytis area. You can see that here they are challenged. Here there is a big wasted area, and here it's much more reduced. The area of damage, it's a modern elite variety, so everyone is genetically identical. The surface destroyed by the fungus is 2.5 more important when there is no mycorrhizae. And when you look at the induced defense, I mean the induced defense, not the basic level of defense. For example, this protease, which is just killing the enzymes of the fungus, so it's a protection. You can see that it's a matter of the way the defense are induced. It's not the fact that there is a higher defense, that simply the immune system is reacting faster and stronger. So in a way, shaping a correct immune system in a plant means that they met a fungus. It's in no way something that could surprise us, because we know now that animals, as long as they don't have a gut microbiota, their immune system is not switched to adult, fully efficient stage. So we have the same with our gut microflora. It's quite a general feature that at germination or birth, you meet the microbes, and this is a switching signal to adult level of the microbe of the immune system. And to finish that, here you have the way different alert hormones are inducing PIN2 as an example of the defense. And you can see that when it's not mycorrhizal versus when it's mycorrhizal, jasmonate, which is the signal, the other ones, they don't elicit anything in terms of PIN2, but the elicitation by jasmonate is potentiated by the fact that the plant is mycorrhizal with this or this fungus versus a non-mycorrhizal fungus. We still don't know more why the plant is more responsive to its own alert signals. We still don't know that, but I would like to offer you a perspective. It's been recently described that in the Mycorrhizal Association, a lot of effectors, small separated proteins, when I mean a lot, it means hundreds, are just moving between the partners to the fungus from the plant and to the plant from the fungus. Here, this is a nice work by people from Nancy in Ray, where they labeled this small effector MISP7. And you can see that this, of course, effector protein is peptide, I should say, is accumulated in the fungus, but also migrate to the nucleus of the host cells in the mycorrhizal association. So it's not only in the fungus, but also in the nucleus of the host. And in this very nice work, they show that it's switching off the jasmonate answer. So we see that small peptides, small separated peptides, they are able to modify the partners. And I would not be surprised if it was one of such mechanisms that would explain how the immune system is enhanced systemically. Whereas here in this example, it's rather switched off to tolerate the fungus. But clearly, we go beyond the association. We have an intimate mixing of regulatory peptides, and this may explain a large diversity of impact on the plant. I shall come back to the mycorrhizal association, but you can see that it's pivotal in plant defense and nutrition. I'd now like to shift to the leaves, because we very anciently were aware that the roots are colonized by microbes. And in a way, we are not very surprised because the soil is full of microbes. But the same is true for leaves. And those leaves are microbial, not only because of the plastids and the mitochondria. The architecture of the plant has a feature, which is an unusual feature for animals, that it's made of a lot of pores. There are pores more or less developed between the cells, and those pores, they are allowing a third circulation, which is very overlooked. We think of the xylem sap and the phloem sap, the raw sap and the elaborated sap, but we don't think of the circulation of the gaze. And the gaze, they just go through diffusion. And those walls, they have connection with external parts by way of stomata, of course, but there are also such holes and such iron chyma, that's the name of this air circulation system between the cell in the older plant, and it's even in the bark, there are some lines of cell, which you can see, for example, on cork of wines. Those lines of cells are very separated, and they just allow the air to flow through the bark to the living tissues that are inside. So really, this system is open to outside. And it makes that where the human or animal tissues are not or very, very poorly colonized by microbes, the tissues of plants are colonized by microbes by way of this entrance, I'd say side by side. So I just, for French-speaking people of you, mentioned this. That's my chronicle, my last chronicle in Médecine Science. Sorry, it's in French. I was devoting it to the iron chyma, an overlooked system of exchange within the third, actually, with the xylem and the phloem circulating system, which is fully overlooked. And read it, you will see that you will realize that it's fully reshaping the plant in terms also of hormonal system. And it explains why plants, they look like speaking to each other. But well, I will not discuss this. It's just a teasing. I come back to that. Those openings are placed for fungi to enter within the hole between the cells. And so the colonization by fungi is a cell-to-cell colonization. It's each plant cell is close to a fungal cell, or even, I don't have specific pictures for that, but also bacteria are entering, and the number of bacteria per gram of living leaf is quite enormous. So you can say that the tissue of plant tissue is an ecosystem, which is not true for an animal tissue. Okay, those inhabitants, they do protect the place where they live. And I will show you two kinds of protection. One is a rather funny one. It's the team of Redman in the States who was publishing this tolerance of Decanthelium lanuginosum to thermal soils in Yellowstone, where there can be up to 50 degrees emanations from time to time. This plant is filled with a curvularia fungus, and well, you can cure the fungus when you are at low temperature. Well, it's low for a Yellowstone plant, okay? You don't have a lot of impact on the growth, but if you go above 40 degrees, then the plant doesn't survive or hardly survives when there is no curvularia. The funny thing is that curvularia doesn't grow in the lab over 35 degrees. But they both survive well, and it's believed that most of the story behind is due to the fact that being a fungus or a bacteria, by the way, living in tissues of plant where the plant is using free radicals derived from oxygen as a way to fight against microbes, means that you should detoxify them. And the free radicals are a bit of an accident occurring whenever there is a stress. So the current view of those, there are many such protections described. The current view is that probably the endophytic fungi or bacteria, since they have a way of detoxifying free radicals, they avoid all the over accident due to free radicals after a stress is encountered. So you have the stress, of course, but you don't suffer from the free radicals that are produced during the stress because they just alleviate this. Okay. I have another funny story that shows that, by the way, neglecting why plants are microbial on how plant or microbial ecosystem can be damaging to agriculture. You may know the story of Kentucky 31. No, it's a funny story of a selection by Kentucky universities of a good ground of Festuca arundinacea, growing very well and released in 43 to improve the pastures of America for the animals, the cattle, to grow better. And the result is that it's growing very well, huh? But it was a catastrophe because then the animals grazing on that, they lost their extremities due to a bad circulation of the blood. They have bizarre behavior. They have, especially they're active in the night, not more in the day. And during the day, they look for water to stay in the water. That's very bizarre behavior. And you have a lot of loss of meat, of calves, of milk production. The outcome per year of the introduction of this grass is awful. And looking for what happens with that is simply that this plant is highly toxic because it's filled with a toxic fungus belonging to Neotyphoidum genus. The fungus grows undefeatically and it's very rich in alkaloid. Some are LSD related and they modified the behavior of the calves. Some are making blood vessels shrinking and they impede the circulation to the extremities. Other are against insects, other are blah, blah, blah. So the plant is growing well because it's just avoided by pathogens. But it's not very good for the animals to eat that whenever there is only that to eat. This undefeated growth is not conspicuous because there is never sporulation. The fungus just propagates. It's colonizing the meristem and it's propagating to the seeds. And the reproduction of the fungus is not achieved by horizontal transmission, but just by vertical transmission. And what the poor guys did, selecting for Kentucky 31, is selecting for the most toxic fungus, since toxicity was inherited, actually. And it's now a nightmare because it's an invasive species, still sold for greens. Because green, you know, they have to be green for golfers to play on the greens correctly. So it's still on sale, whereas it's an invasive species. Here you have the choice of small aphids of Lolium multiflorum with or without the underphyte. You can see that there is a choice, and it's due to pyramine, this compound that is a very anti-insect efficient compound, an alkaloid which is just making the aphids to avoid the plant with the underphyte. And that's it. So it's quite common that there are such partners. And it's also quite common that they are vertically inherited. Just something from a Psychotria, a rubiaceae, that is a genus of rubiaceae that has some leaf nodules that were long believed to be nitrogen fixing. They are filled with bacteria. Recent work by Julien Carlier and others, they just showed that it's just bacteria producing anti-insect toxins. But this is vertically transmitted, the Basis bacteria. They colonize the meristem, and from there they colonize the seed. Okay, so the leaves, they are also colonized, and the role for nutrition is not so high, but there is a role for protection. Just a short story now of something on which you will not find any paper. It's not published. There is no one knowing that, but some mycologists, and there is no data behind that. These trees, they look normal for you. They have a trunk and branches. You remember that they were small. When they were small, they had branches from the bottom. And now they grow older. They are bigger in trunk. Where are the branches? Gone. I don't see them anymore. Where are they gone? They may stay, by the way, if there is light. If you are not in the forest, the branches, the twigs are from the very bottom. So you will tell me, okay, they went to death because there was no light. It's not exactly true, this story. Let's take introduced plants such as Cupressus or Pinus insignis in Brittany, or Douglas fir everywhere in Europe. The lower branches are still there. They are about to be dead because they lack light, or they are dead because they have no light, but they are still there. And by the way, it's a drama for many of those introduced plants. You have to prune them. Even Quercus rubra, a quercus from America, you have to prune it. Why? Because the microbes doing the job are not introduced. The pruning of the lower branch, whenever laking light, they are weak or dead, the pruning is a microbial process. There are microbes killing or eating those branches. And there are a big rule for saprophyte is that, for saprophyte of wood, is that there is specificity to the size of the wood block. Because once they need a lot of oxygen, they will never invade the trunk itself. Because the wood inside, there is not enough of oxygen. So they eat the branch and they go to hell or to sports. And after that, there is cicatrization of the trunk. Sorry, it's another one. Cicatrization. And after cicatrization, you end up with a fiber that is more continuous, so trunks which are more resisting the mechanical stress, and a closure, a full closure of the bark, which is avoiding the penetration of parasites that could attack the big block of woods inside the trunk. So it's a very important thing for health of the plant in terms of mechanical resistance and sanitary state, but it's very overlooked. And believe me, behind mycologist who knows the species doing this ecological service, there is no work on that. Except the fact that forester, whenever the species are introduced, they know they have to prune the lower branches to get nice trunks. That's the only thing we know about, the only research published about that. So now let's finish with reproduction. First, funny fact about the flowers to start with. Part of the attractivity of the flowers is microbial stuff. You can, for example, read this nice paper on Sambucus nigra, which is a plant that is nicely smelling. There are a lot of recipes of syrups, wines, or even distillate based on that. It's rich in terpenes. And in this experiment, they have a control which over a big week is producing those volatile terpene, and in an amount that is finally getting lower, of course, because the inflorescences are getting old. And now another bunch is submitted at day 0, is submitted to antibiotic fumigation, and the production of terpene is crashing down. The nice thing is that the precursors in those red flowers are accumulating. So you end up with a story that is, and the same is described for vetiver, if you know this aroma, this plant producing an aroma that is very much used in, for perfume production. The precursors of the smell are produced by the plant, but the volatilization is due to microbial modification of the precursors. So it's funny. It goes farther. Among the endophytic bacteria that are vertically transmitted, you have those methyllobacteria, which are eating on methane and methanol issuing from demethylation of pectins. And those intercellular bacteria, again, colonizers of those ironchima areas, they are involved in cytokinin secretion, and exactly as serotonin for humans. The plant can do cytokinins, but not in an amount that is sufficient at some point to fulfill the functions. And here you have the germination of Glycinemax. So some of these papers are quite old. This is why there is no standard deviation. But it's just that it's stories that were not, they are known and no one replicated them in the area where we publish with standard deviations and so on. So I'm sorry, but it's like that. Anyway, you have here the germination with spontaneous bacteria in the seeds. Now, when you enrich in this bacteria, it germinates more often. When you kill the bacteria by heating, it's very easy to get rid of some of the microbes of the seeds by heating, because the seeds, they survive the heating, but not the fungi or the bacteria. So then you reduce the germination. You can come back to previous phenotype. You can compensate by adding the bacteria or adding cytokinin. And it's true that they produce cytokinin, and it seems that at germination, there is a need of cytokinin that is partially depending on their presence. So it goes to germination in terms of reproduction. And by the way, reproduction is the time of the life of the plant where few of the bacteria and fungi may be transmitted to the offspring, so that the equipment is not necessarily gained from the environment horizontally, but it's sometimes vertically transmitted. Okay. We end up with nearly no function in which there is no contribution of microbes. And by the way, the same story happens with animals and their microbiota. So it seems to be pervasive. And I would like, for some minutes, to put a backbone to that. Maybe it's the most important part of the talk, I think, because that's the question: why the hell are the plant and animals not able to do it alone? It's so important to reproduce, to defend yourself, to feed. Why no autonomy? And I would like to show you that symbiosis, which is often described as the emergence of new properties and as a complexification, is sometimes a complexification, but here also the door to dependency and simplification. Let's imagine two species, one, let's call it gray species, and the pink species. They are living in the same environment, okay? And we will study the standing, the ability to stand up. Here you can delete one of the species, the other one is standing. They are autonomous. Let's imagine they are always side by side. It means that they have a symbiotic relationship, or they are both parasites on the same thing. They are always together. Still here, I can delete this one or this one. The standing, the function of standing is achieved. But whenever you are always with this coexistence, such a mutation is possible. Don't tell me it's advantageous because it's an economy of carbon. The leg is still there. But it goes with that, okay? Simply because there is a kind of redundancy. Now the standing is a property that is in the two partners, and one can lose it without any problem. There was no positive selection for that. It's just drifting. Who has the responsibility for the function is neutral. It's a neutral contingent evolution, and it's often irreversible because there is maybe a modification here that will be difficult to circumvent, and there is no selective pressure to come back to an autonomy. And so much of the dependence you can see now are just due to the fact that by coexistence, you are driven to loss of autonomy and dependence. It's often symmetrical. It can stand like that, okay? It's and by the way, it's a way of publishing a paper where your picture is 5 times in the paper. That was one of the goals, and there was yeah, there was a bait behind this story. But okay, anyway, it's a kind of ratchet mechanism where step by step you lose autonomy by way of all the coexistence symbiosis is offering. So not only the dependence comes from the fact that sometimes you get a new property by getting a partner, but also it can simply result from the fact that you lose autonomy and go into dependency by way of this mechanism. I don't oppose them. They are just complementary in making the false good idea that plants and animals are holobionts. This is a shitty idea if you think of it, because you realize that the plant and the animal are not able to stand alone. So what do? You put the microbes which are necessary in addition to that, and you close the box. You make a new autonomous entities. Not only I can predict to you that there are some dependents that are not embedded in the holobiont, but I think that it's missing the point. You still look for autonomous entities and functionally delimited systems. Whereas the story here is that the link between species, the relationship between species is relevant to know how species are functioning. You can try to run for a new, but without fungi to decompose something in the soil, this will not grow. You can make a holobiont, you can call me a holobiont, but if there is no food to eat, I will not survive. So it's time now to enter in the idea that we should less or not only study organisms, but the link between organisms as one of the most important focus of biology. How nowadays biology is like that? We study species 29, species 30, but unless you don't see the link between the species, you will not see what you study. Okay, now what to do with that? I will not try to cover all the steps or the point of physiology of the plant which we could address, but I would like to focus a bit on mycorrhizal stories. I would like first to summarize something from those two very nice books that were published under the lead of Lionel Ranjard and Inray of Dijon, the two atlas of soil fungi and bacteria of France. The take-home message is the following one. If you look at the quantity of microbial DNA in soil, you can see that in all the agricultural regions, the blue ones, it's correlating with a minimum of microbial biomass. And the microbial biomass is decreasing with increased management. Forest, grazing place, culture, wine, and orchards. Monospecific orchards, of course. If you now look at species diversity, clearly the driving for bacteria, at least, or for fungi, by the way, clearly there is not a lot of variation, and the driving mechanism is not the farming, because this doesn't correspond to any farming practices. And the same for the fungi. It's more corresponding to other climatic or soil properties. So it means that the good thing is that we are in a situation where our soils are not dead and the species diversity is still there. Simply the number of individuals is collapsing, which is the door to extinction and also the door to lower resilience by lower genetic diversity. So there is a problem, but in terms of number of partners, of microbes, sorry, but the species are still there, so it's time to wake up. Now, one way of waking up would be to inoculate the good guys in the soil. And especially I would like to focus for some minutes on, shall we go to, the name of the company is not visible here, to buy all the things which promise us to inoculate mycorrhiza in our fields or in our gardens. Shall we do that? Before that, we have to consider this very important paper by John Chironomos in 2003, where each line is a different species. And on different species, he was inoculating one strain of Glomus etunecatum, one of those fungi performing mycorrhizae. So this fungus is offered to many plants. Those plants, they can grow alone. This is the 0 in sterile soil, or with the fungus. And for most plants, we see that with the fungus, they produce more biomass. For evolutionists, it's meaningless, but for agronomists, it's very useful. An evolutionist would have liked to see the number of seeds, of course. But for agronomists, it's very nice to see that it's improving the growth, as I told you. But for some species, it's not. And it can be up to 40, 50, 49% less biomass after inoculation. It means that for mycorrhiza, it's like for couples in human normal life. It's just a matter of being two. It's not just a matter of being two. There is more than that. There are some cases where it runs and makes the best, and some cases where it's just a way to the hell. In other ways, I want to stress out that whenever you inoculate a limited number of strains, and very often in those inoculants there is 1 or 2, is it good for your strawberries? Is it good for your wheat? We don't know that. It's not written on the, just written, put a bit of water, dilute it in water, and try to introduce it under the full moon, because otherwise there may be some bizarre effect. There are a few papers like that, and this one is funny. It's a Canadian team that is very known to have some very good mycologists, and they discovered funny things. They were using a set of products that had 12 species declared, and what they found is that in the whole trial, they found 5 species that were not declared, and 4 were not visible, and 4 declared were not visible because they were not able to recover the evidence, the spores, the evidence that they were there. And more funnily, 6 weeks after planting, only 6 of 7 inoculants had a slight increase of biomass. It's just because when you do anything, anything happens. C'est en faisant n'importe quoi, on devient n'importe qui pour French-speaking part of the audience. In another way, we see that probably we face this problem. And very often, the result is not very good because first, it's not adapted to soil. By the way, not any fungus goes in any soil. Second, it may not be adapted to the plant. But there is more. By the way, those fungi are very often exotic. And do we want to introduce exotic fungi in our soils? By chance, it seems they don't succeed much in settling and so on. But the source is not local, which may be bad in terms of invasion. It's not soil adapted, it's not plant adapted. The result is very unsure. It can be sure. And some companies, like this company, I make it clear, I'm discussing that. I'm at the scientific advisory board of this company, but I earn nothing, okay? Let's be clear. So there is a potential conflict of interest, but not in terms of money, just in terms of fun and reputation. So what they do, and they're not the only one to do that, but for them, I can access the data and show them to you. They go to your field, they amplify by way of a patented mechanism, but briefly, they have a good mix of plants that enhance the sporulation of many diverse fungi. They enhance your fungi and they give you back your fungi, which are adapted to your soil. And they even offer a last round of selection where they select it on the plant you wish to plant yourself in the field. And what you see here, it's on grapes. You can see that the production with their treatment compared to the control, it's a field control. It's in a vineyard, not inoculated. And you can see that there is more fruit produced because there is more clusters of fruits. And each cluster is bigger. So they get a result. To be honest, you shift from 30% positive result with commercial inoculum to 70. You don't shift to 100% positive results at all. But sometimes you obtain that, and on those grapes, they had an opportunity to observe what happened in a year of big drought. On that year, there were less evidence by means of carbon-13 of drought stress in mycorrhizal versus control plants. Control being mycorrhizal but not inoculated, okay? I should write inoculated here. The soil had enhanced water retaining properties because of the external mycelium, which is contributing to the soil properties. The root growth and the yield were increased compared to the control. Here it's another experiment they did. You have controlled soil, so naturally mycorrhizal, commercial inoculum, and indigenous fungal inoculum enhanced by the mean of preculture and given back to the soil. You can see that there is no difference between commercial and control, while with the more specific inocula, they get a longer time of fruit production with bigger fruits. It's when it goes, but once again, as in many solutions based on nature, you are not very sure it will work anytime, but it's working more often because at least you have local adaptation and adaptation to the plant. This said, you are not finished there. You need also to have good practices that will, as much as possible, allow the fungi you settled to stay. What am I pointing out? First, mineral fertilizers. It's well known that the more you put fertilizers, here this is wheat field fertilization, and here this is a high garden fertilization in phosphorus. You can see that the mycorrhizal level is decreasing when fertility is there simply because the plant needs less fungus. If you look at the impact on growth of those different fertility level, when there is no fungi, you can see that the more you add phosphorus, the more it grows. When there is the mycorrhizal fungi, and this is the condition in which you see that the mycorrhizal level is decreasing with phosphorus addition, you can see that the more there is phosphorus, the more it grows. But you shift from a situation where the fungus is a good news in terms of growth to a situation where it becomes a load because it's still eating a part of the carbohydrates of the plant, but it's not providing anything in terms of phosphorus. but still it probably has some rules on stress or pathogen resistance. But that's another PNAS paper that showed that when you put minerals fertilizers, you reduce the amount of microbial fungi in the soil while you stay with the same pathogenic load, of course, because pathogenic load is poorly, at least on short term, reacting to the fertility. By the way, you can see that here, mineral fertilizers are just trying to need more defense and possibly chemical defense of the plant because they are less protected. The opposite happens. Many of the pesticides are having a negative impact on microbial fungi. And one of those, and it's quite unexpected because it's something that is normally killing the plants, is glyphosate, which is highly toxic to the spores of mycorrhizal fungi. And that's a problem that you end up with killing the fungi by glyphosate. So you need more fertilization because you will rely on less availability of fungi to feed the plant. So we've been feeding the world in an agriculture where with mineral fertilizers and pesticides, we've been leveling, buffering the variation of the production. The quantitative problem was solved and there is no more problem of hunger, or it's based on political instability and so on. But fertilizers, they whip out, they make a lower need for microbial fungi. Pesticides, they kill part of the inoculum. So you rely less on the microbial interaction. So you need more help to defend the plant and more food to feed the plant because you have less of this interaction. That's the story on how we solve the quantitative problem by creating a kind of dependency to additional functions or to additional practices that are just doing a job that was done by the microbes before. There is another problem with tilling. Tilling is a very efficient herbicide because it's destroying the roots of the plant. And after all the rhizomes and after tilling, you don't have any plant anymore. But you may realize that it's the same for mycelia of fungi. They are just killed by tilling. And after tilling, some fungal species do not survive. So it's killing the fungi. And finally, it's not killing the bacteria. Not only it's not killing them, but some species of bacteria are present after tilling, which are not present before. But if you think of it, tilling is oxygenizing, putting oxygen in big amount in the soil. So more oxygen demanding bacteria, they survive better. Plus, there are less competitors on the fungal side. So it explains that the bacteria, which are poorly disturbed by tilling, they can proliferate, and even some species can join the soil, which were not there before. So it means that in terms of good practices, excess of mineral fertilizer is not good, excess of pesticides is not good, but also tilling is not good. And when you think of that, this is a dual killing of the fungus, because tilling has been destroying the fungus, the fungi, so you only rely on the spores, but there is nothing to eat for the fungi, so the spores, they don't develop. You don't get mycelium, and you will wait for your next generation of plant to wake up the spores and re-establish the mycelium. That's a good winter where you sow, if you can till or not, but when you have tilled or when you don't till, you can also, it's a different thing, set up cultures and mixed culture that will enhance the development of mycelia of microbial fungi from diverse species, each plant species, allowing the development of one fungal species. And as a result, you will have an established mycelium network with various species for your next culture when you will have destroyed this cover. So those are perspectives. Now, what to do? What are the next step, and what kind of agriculture do we have to imagine to go with at least this mycorrhizal interaction? So you may inoculate, but be it inoculated or not, you need the good practices. And if we take this, the good practices is using the plant to boost the microbes associated with roots. And if you think of it, we've been doing that since 5,000 years. If you look, Columella, for example, the Roman agronomist, he tells you how to sow regularly legumes. And the fact is that whenever, sorry, it's a bit not in English here, translation was not total. If you look at the rhizobia that are the nitrogen fixing bacteria associated here for this species to clover, to vetch, help, luzerne?
Alfalfa.
Alfalfa. Okay. So you can see that in the rhizosphere, there is a lot of those nitrogen-fixing bacteria released from the root, but they're also in bulk soil, whereas if you sow barley, they are hardly in the soil. So we've been handling the microbes since millennia by handling the plants that associate to microbes. And I think this is a source of inspiration. We've been using those bacteria and enhancing them by way of just cultivating their host well before we knew nitrogen and well before we knew bacteria. And I think that for now, better than inoculating or making analysis of soil microbiota, we have to use the plants. Because with a permanent cover, we permanently feed the fungi. And there is no place, I think, for bare soil in our ecosystems. It's more than tilling. It's just this idea of feeding during the bad season the mycorrhizal fungi. By the way, this kind of, there was a name given earlier, isn't it? Well, what you call catch crops? I think it was given today as a catch crops. Yeah, okay, so I didn't know the English word, so I was happy to listen to that, and I was not sure of the translation. Catch crops, they are good in many other respects, like against erosion, like bringing more carbon to the soil when you will destroy this cover, or bringing nitrogen. So they have many positive impact, but they are pre-cultivating the fungi of your next culture. And here, there are a few papers dealing with this issue, how to foster the next generation's mycorrhizae, but this paper at least is doing it. We will sow maize on fields that the year before received, so we are in southern Japan, so here you can have 2 harvests per year. One is not very good, one is good. So you have been having either cabbage, then fallow, or fallow, then not wheat, or cabbage, then sunflower, or fallow, then oat, or maize, then fallow. It's red when the plant or the mix of plant is mycorrhizal, and it's blue when it's a non-mycorrhizal plant. And now we sow our maize. And what do we see? That's the number of spores. We see that whenever there was a non-mycorrhizal plant, you tend to have lower number of spores of mycorrhizal fungi in the soil than when you finish with the sunflower after cabbage, or when you have 2 generations in the previous year of mycorrhizal plant. Now, what is the impact on the maize? At 2 points, the mycorrhizal rate was evaluated, and you can see that it reflects the number of spores. So some cultures were making few spores, and it results in few mycorrhizae because you rely on a few number of spores. Whereas when you had some pre-culture that are encouraging more or a lot the mycorrhizal fungi, you get to a higher mycorrhizal rate. When you look at the biomass of the plant or the grain production at harvest, you can see that there is still a positive impact of some of the culture of the previous years, and that whenever you have some succession involving a non-mycorrhizal plant, you may have a lower yield. Now, it means that what you cultivated before is a way of influencing, by way of the fungus, what will happen later. This team produced more recently kind of many permutation of pre-culture, post-culture, and they offered a way over 4 years of having a succession that is the most, it's over 4 years only. One may hope longer succession of culture before turning back to same culture, but here they offer a way over 4 cultures to improve significantly the level of mycorrhizal association of the plants. I'm not sure this result should be taken as a picture to do it here. It may be very dependent on the soil and the climate, but in each region, there may be a way of optimizing what's going next by way of either the choice of what we do on 1 year on the next year, or by using the catch crops as a way of enhancing the system. And you will have to do that even if you pay some inoculant once, because it will help. By the way, one way of maintaining inoculants is agroforestry, because not only the trees, if you choose them well, but also the herbs under the trees that you don't fully destroy, they are mycorrhizal. And in this nice paper from colleagues from INRAE in Montpellier, you can see that the mycorrhizal rate and the arbuscular, it's the intracellular symbiotic structures. The mycorrhizal rate and the number of intracellular colonization by the fungus is higher on the wheat close to the herbs and the walnut than far out of the influence of the row 3 meters away of the row of trees. However, if you read carefully this paper, you will see that probably the most inoculant effect is not due to the fungi associated with the tree, but to the fungi associated with those herbs. Anyway, the trees in agroforestry may also be a reservoir, a reserve of fungi to colonize the plant. Here, I show you a couple of work we did in a system of walnut plus maize. And you know probably that maize is a C4 plant, while walnut is a C3 plant. So the carbon-13 content is not the same. Now let's look at the mycorrhizal fungi, okay? The fungi on the maize and on the walnut are intermediate in carbon-13. It means that they are fed by the both partners. And that's the functional evidence that not only the maize are a bit feeding the fungi you find under the walnut, but also the fungi you find under the maize is fed a bit by the walnut. And it's a kind of functional evidence that we have a trafficking of carbon, and that the fungi we find in the maize, they partly rely on the trees for their nutrition. So it goes further in the idea that having trees, you inoculate and maintain a mycelium that may be useful for the annual culture. And it's another reason to think a bit more of hedgerows and to investigate them, but there are, to my knowledge, very few data on that, on how they are a reservoir of microbes helping the plants. And I will now say microbes helping the plant because I showed you a few data on microbial fungi, but there may be some other microbes in the soil, fungi, which are not mycorrhizal, or bacteria that may be helpful and not specific enough to be supported by one species and then reused by the target species in the field. I hope I convinced you that this is microbial and that it looks bizarre to say it like that. And if it looks bizarre, it's because we never faced that conceptually. And whenever there is something you did not face conceptually before, there is probably much more improvement to hope than in the things you conceptually were facing before. And that's the hope that we can do something better. For those of you who are already French, there is a book to read, perhaps. I wrote on those issues. But more generally speaking, I'd like to thank you for this bringing ideas together, because I was myself learning a lot from the 2 time I spent there. And I think it's very nice to have such a diverse audience. And I finish by the point I should have started with, to thank you for the time and organization.
Thank you for coming.